Dear student, welcome in class of plant propagation and nursery management for fruit crops. Today we are going to discuss about the various, you can say, asexual reproduction methods related applications. So we are going to discuss about the application part. What is the role of apomixis? What is the role of tissue culture? What is the role of different vegetative propagation techniques? So we know that apomixis involves the development of embryo by the asexual means as a replacement of the normal sexual reproduction. So, which may be associated with failure of meiosis during megasporogenesis, which is related to initiation of mitosis in unfertilized egg cells and the other diploid cells of embryo sac. In case of meiosis that has occurred, there may be same phenomena, but if the embryo sac which has resulted in meiosis and is being involved in apomixis process, it becomes non-recurring kind, as there is no survival of such kind of embryo or plants developed from such embryo. So gametophytic apomixis, we know that it is related to the apomixis which is through the gametophytic cells, while the sporophytic apomixis is related to that of sporophytic cells. So it is controlled by, gametophytic apomixis is controlled by the dominant gene or sometimes the linked genes on the same chromosome, and apospory is also due to dominant genes and has been reported in ranunculus species. So nucellar or adventitive embryony in citrus is controlled by single dominant genes. So you can understand that almost all apomixis process is related to the dominant genes, may be influenced by some linkage or may not, as per the different species. What is the application of apomixis or nucellar embryony that we have to discuss here? So it is going to involve in the fixation of heterosis. So we know that heterosis is what? It is the superiority of F1 hybrid over the parents. So if the F1 hybrid is superior, and we know that it is bearing character of both the parents, they are dominant as well as recessive. So when it is going to, since it is heterozygous, which undergoes segregation, if it is being grown or if it is being reproduced sexually. So what is that? Apomixis brings the fixation of such kind of heterosis in the population. So if there is obligate kind of apomixis, means there is only one method of reproduction, that is the apomixis. So if it is obligate apomixis, it results in complete fixation of heterosis, but if it is facultative approach, then it causes the partial fixation of the heterosis, like in Bahia grass and buffalo grass. So how it is going to be done? It is you are going to cross sex, you can say male parent, which is a sexually reproducing line, with the female parent as apomictic line. You will get the apomictic hybrid, and it can be easily maintained and multiplied through the seeds. So that advantage is not possible if instead of apomictic line you are using any sexually reproducing line as a female parent. So here it is very good if you are going to utilize this apomictic line as a female parent, you are going to fix this in the population. So the second application is in order to produce homozygous line. So some types of apomixis are going to be involved, and because it is uh, having par parthenogenetic development of egg cells, and followed by the development of embryos from secondary diploid cells of embryo sacs that is derived from the fusion. So what is that? You are going to develop homozygous lines here. You are not going to develop some cells through the fusion or fertilization between two different species or two different individuals. So it is the complete involvement of inside the embryo sac, that is the development of diploid cells of the embryo sac, that is the result of the fusion, and followed by the development of embryos. So this is what you can say a good way to produce homozygous line. Further, production of hybrid is also very good when you are going to use of two facultative apomictics lines. So they reproduce and maintain. If you are using the seeds, you can maintain it through that, and the seeds can be grown in the every generation, which is generally not possible into the normal F1 hybrid. So example is the sorghum. Adventative embryonic for uniform root stock, we can develop the virus-free sign. We can develop virus-free root stocks by using the adventative embryonic concept. So this is one of the advantages. So it is a good way, and it is now being greatly applied. Either it is introduced apomixis or it is the natural apomixis. So to maintain the superior genotypes as a clones of seeds, which is not possible through the normal development of seeds as a sexual reproduction. So this is the advantage, and the recent, you can say, application. So advantages of apomixis includes the same thing that we have discussed in the application. So it is the permit fixation of heterosis in the hybrid. So the seeds can be sown again and again after harvesting, and the new hybrids can be multiplied from the small number of hybrid seeds to develop a large amount. So apomixis also provides the means of commercial production of F1 hybrids in crops where there is a CMS, cytoplasmic male sterility, or there is a situation of fertility restoration where such kind of things are not available. So important thing is that you are going to produce F1 hybrid commercially with the if there is no normal process of hybridization, where you can say the normal process of hybridization is very difficult because of lack of CMS line, lack of fertility restoration CS system. There are some disadvantages also, and that is the tedious and time consuming process in case of facultative apomixis, and proportion of sexual progeny is affected by environmental factors also. At the same time, the genetic base is not clear, and there is a complete absence of morphological markers linked to the apomictic development. So the maintenance of apomictic stock becomes very difficult. Now, how to screen apomixis? Because we know that apomictic seeds or embryo is having, you can say, the seeds are going to develop so many apomictic seedlings, as well as there is a development of zygotic seedling also in case of polyembryonic, and so it becomes very important to identify apomictic seedling to allow its growth. So how we can go, we can go for that? So it is the psychological analysis of embryo sac development, cytological, sorry. So we can analyze the embryo sac through its cytology, chromosomal movement and all these things. Another method is Callow's fluorescence techniques in combination with the clearing of ovules. So it can be used to identify the diplospory situations, as well as the uniformity of the progeny from the cross-pollinated parents is the best indicator that can be used. And the other method can be as a maternal phenotypes among the progeny of F1 crosses. So there is no or limited genetic variation in F2 progeny, high speed fertility in aneuploidy, triploid and white crosses, and multiple seedlings per seed. So these are the things which we can utilize to identify further DNA marker analysis. So you can go for this, which is one of the best and you can say most scientific technique to identify the apomictic seedlings or apomixis situation. Another method that another application that we have to discuss is plant tissue culture related. So we know that micropropagation is one of the tissue culture technique which is used for vegetative multiplication of ornamental plants or fruit plants. So you can produce very large number of plants from the very small, you can say, plant materials such as explant, and each of these plants will be the clone genetically identical to the original parent. So that is what we can do, and the tissue cultured plants can be taken out from the culture and can be grown in controlled conditions and further hardened for transplantation. What is the advantage of that? Is the rapid multiplication takes place within very small time and a small space, and the plants are obtained under the controlled condition, which is possible. This propagation is possible in any season. So the sterile plants which can't be reproduced sexually can be multiplied by these plants by this method as well as. So the rare. So it is also used to conserve the rare plants or endangered species. So it is used to develop the virus-free planting material, most commonly in potato, sugarcane, banana, apple, and many more. Large scale growth of plant cells in liquid culture is also one of the good technique to which is used in bioreactors for production of important compounds. So you can say medicinal use, you can say it as. So cross of distantly related species by protoplast fusion and regeneration of the novel hybrid is only possible through tissue culture techniques. So development of stress tolerant plants related to salt tolerance, drought tolerance, heavy metal tolerance, low temperature tolerance, herbicide resistant. So these all can be developed rapidly by the in vitro selection of the stress tolerant plants. So even it can be utilized for the regeneration of the genetically modified plants, which is one of the way to add some genetical traits in the population. So this is what the good application of you can say tissue culture technique. In addition to that, vegetatively propagated plant and its you can say method is also quite utilized in the different purpose in horticulture, that is development of two to the types planting materials as well as multiplication of species which can't be produced through the seeds, so they can be produced, they can be multiplied through the you can say vegetative propagation method. And at the same time development of clonal root stocks is are also possible, and these clonal root stocks are having very good application in the propagation techniques. Further fixation of hybrids also is possible through this because hybrids if they are going to be produced through sexual method, they are going to segregate But if it is propagated vegetatively, the segregation cannot be possible. So it is one of the good way to maintain the hybrids. And germplasm conservation of plant species is mostly ensured by the common vegetative process. These are some common applications of vegetative propagation techniques.